Several years ago, the NCAD published a vision for reducing the use of animals in procedures, with the ultimate goal, a dot on the horizon, of achieving a worldwide shift to research that doesn't use laboratory animals. That dot has triggered quite a response. As a scientific committee, we are aware of the vital nature of intrinsic motivation, time and funding. Yet, we also know that the true innovation often comes from unexpected quarters. We are convinced it helps to promote those unexpected innovation by sharing target situations for research methods that don't employ laboratory animals in specific research domains. This includes objectives and action plans. The use of animal-free innovation is the central focus, which in the Netherlands is arranged through TPI.
TPI connects a broad range of stakeholders such as scientists, regulators, funders and NGOs to accelerate the rate of innovation. On TPI TV, scientists exchange ideas and knowledge, and the community can help you to answer challenging questions. Do take a look at TPI TV. Maybe you will even find things you didn't search for but which turn out to be very useful.
Our goal is to make that trophy of animal-free research a challenge cup that will be passed around by scientists in every field of research, in every country. Sounds exciting? Contact TPI.
Good afternoon and welcome to the first of two webinars on the 3 R's in COVID-19 research organized by the 11th World Congress on Alternatives in Animal Use in Life Sciences. I'm Brian Maguire, a journalist based here in Brussels. I'll be the moderator for the event today. And on both days, presentations will highlight innovative model systems to study COVID-19 and will also discuss new strategies for the development of vaccines, therapeutics and diagnostics. These webinars are supported by the Netherlands National Committee for the Protection of Animals Used for Scientific Purposes. We thank them for the support today. Just some quick housekeeping. Questions to the speakers can be sent to the chat box. Our team will moderate and aggregate the questions. Please direct those to each individual you would like to answer, or we will try to allocate them as best as we understand. And the hashtag for today is WC11 Maastricht. You also see that on the front of the web page. We have three speakers for you today: Dr. Penny Hawkins, Koert Stittelaar, and Christian Desaintes. They will each have 20 minutes to present, and I will give them a 3-minute warning just so they can wrap up for us. After that, we'll have around 4 o'clock, 4:05, discussion with the speakers, and we'll put your questions to them as well. And then we aim to wrap up at about 4:30 today. Thank you for all taking your time today to be with us, and we hope this is of real value to you. Our first speaker today is Dr. Penny Hawkins, and she's Head of Research Animals Department at the RSPCA in the United Kingdom. Penny, good afternoon. Thanks for joining us.
Thank you very much, Brian. I'm delighted to be part of this webinar today.
So you have 20 minutes, off you go.
I do. Yep, thank you very much. I'm just waiting to see my first, here we go, my first slide.
There we go.
So thank you very much for that introduction. I've really been looking forward to being part of this today. So can I have the next slide, please? Thank you. So just a quick slide about my organisation. We're a science-based animal welfare NGO, and we deal with all aspects of human-animal interactions. With respect to the use of animals in research and testing, we promote the fullest possible implementation of the 3 R's, with replacement, of course, being the ultimate goal. And alongside that, we champion robust ethical review of animal use to stringently challenge whether and how animals are used in research and testing. We work constructively with the full range of stakeholders, including governments, regulators, industry, the scientific community and other NGOs all over the world. So I think it's important before we begin this webinar just to acknowledge the true human impact of this pandemic. Of course, it's causing devastating human suffering and distress on a global scale, and I'm sure we've all been affected, whether it's down to changes in the way that we live and work, to vulnerable family members we're concerned about, possibly to more serious consequences. So the enormous effect on people's lives and livelihoods cannot be underestimated. And of course, there's an urgent and legitimate need to address this. But does this mean that anything goes? I'm to argue that no, it doesn't mean that, and there are scientific, practical, and ethical reasons for this. So of course, people are taking a number of different approaches to try to identify cures, therapies, vaccines, and medical devices to help people affected by corona. This includes epidemiology, computer modelling, in vitro studies, including organs on a chip, clinical trials, including repurposing of existing drugs, which can go straight to phase 3 trials under certain circumstances, and animal models. And it's important to note that nobody can predict the extent to which each of these will contribute to any successful outcomes. Undoubtedly, they will all play a part, but the extent of the contribution is not easy to predict. And of course, within this webinar, we're primarily concerned with animal models and the techniques that can be used to replace these. So I'm going to argue that this current crisis has given rise to both negative and positive outcomes for laboratory animals. So thinking of the negatives first, the first one that springs to mind is the race or scramble to develop new animal models. There's a wide range of species involved, as you can see from the top of this slide. And while any use of animals in research and testing has the potential to cause suffering, unfortunately, coronavirus has the potential to cause severe suffering and mortality to animals used in this research. So here are just a couple of examples from the literature. You can see an example of mortality in older ferrets, which kind of mirrors the situation in humans. And there's also a list of clinical signs that have been observed in hamsters. And you can see these are instantly recognizable as indicators of severe suffering. So we know particularly that dyspnea or air hunger is very distressing for humans and animals alike. So that's one negative aspect. Another negative aspect is the way in which this suffering is often glossed over or trivialized. So here are some examples of ways in which animals have been presented as heroes who are helping scientists fight COVID-19. For example, there you can see the example, fuzzy friends working alongside their humans and thank you mice. And clearly, animals are not heroes and they wouldn't choose to help, even if they could. So this is a negative aspect also. Another negative aspect is so-called desperation science, and this is being widely commented upon. So you can see that the US Department of Health and Human Services vaccine development program is actually called Operation Warp Speed, and it's led to a vaccine efficacy trial, although I see from the media today that there are some warning signs being highlighted associated with this. And while this breakneck pace is understandable, there are some risks associated with this that commentators have been highlighting with respect to clinical trials. So people have been citing low quality studies that lack rigour and duplication of effort. And now if this is happening in clinical trials, it's highly likely to be happening in preclinical trials as well. And you have two issues. Not only is it holding up treatments and vaccines for people, but it's also wasting animals' lives. And here's a quote from a worker in US public health policy, which sums it all up very nicely. Desperate times do not call for desperate measures. They call for evidence-based medicine. And in the case of preclinical research, they call for humane and responsible science. A final negative aspect is the so-called COVIDization of research, which others are also starting to comment on. Here's an article from Nature about this. So everything is rushed. People are beginning to work on COVID-19 who perhaps didn't work on this before, so it's actually detracting from some other research field. And there's so much noise that there's a real struggle now to separate the signal from the noise. And this commentator also raises concerns about dubious and retracted research, lowering of scientific standards, and bad science. And again, this is extremely damaging, but if animals are involved too, then it's a real concern for all of us. So just to pause then, before I come on to the positives, This is kind of a testing time for the present evaluation frameworks that are used to, in the licensing process for animal studies. So we know that there are thousands of research projects worldwide that are associated with COVID-19, and we know that many of these are going to involve animals. And there are a number of different project evaluation processes in different legislations, for example, the harm-benefit analysis and the whole project license application process that's set out in UK and European legislation. And it's clear within this that project evaluation should not just focus on the importance of the overall problem, and clearly this is an enormous problem, but it should focus on what the specific project and the specific use of animals will contribute in terms of value towards that project's specific goals, and do these potential benefits justify the likely harms to animals. And this has really got to be robustly adhered to, even in these desperate times. And it can also be viewed as a kind of a test of the ethical framework. So thinking again about UK and European legislation, it's accepted that higher likely harms can be justifiable if there are higher potential benefits. And also within the EU directive, there are various safeguard clauses. So for example, there's one that could allow great ape use in a situation such as this. Although I have to say, I've not heard of any proposals to use great apes in coronavirus research. And I've just put a copy of the Bateson decision cube there, which I'm sure you'll all be familiar with. And I've highlighted the importance of research there. So clearly this is important research, but that's just one factor. It's also essential to consider the likelihood of benefits and the animal suffering involved. And that really has to be used robustly. It's important that the high importance doesn't skew the rest of the decision cube. And certainly within clinical research, there are voices of reason. So here's Professor Andrew Pollard of the Oxford Research Group, who's saying there's no justification for changing the ethical standards. So he's a member of the UK group that's been making good progress with identifying and developing a vaccine. And he said they go through exactly the same process for their research projects now as they have done for everything over the last 25 years. The only difference is that because it's so urgent, the ethics committees are readier to review the information in a faster timescale than they had been before. So that's a voice of reason from within clinical research, and I hope that there are similar voices of reason within preclinical research too. So on to some positives then. One positive is raised awareness of animal use. So I cited earlier some irritating and frustrating coverage that suggests that animals are helping heroes. But amongst that, there's also increased awareness of the price that animals pay currently for medical progress. So there are a lot of headlines like this which have been raising public awareness. Next slide, thank you. And as we know, public opinion polls have repeatedly shown over the years that people have very legitimate and real concerns about animal use and suffering in research and testing. And even at this current desperate time, they are still concerned. And a new survey has shown that people feel conflicted about animal use in coronavirus research. And there's a quote, "Because I can accept it doesn't mean that I like it." And that's a good thing. That public concern is legitimate and justified, and hopefully it will also help to drive replacement of animals. Another very clear benefit has been the unprecedented sharing of data and collaboration and publishing of research outputs. Also, there have been a number of global health bodies that have revitalised and re-endorsed a global statement on data sharing in public health emergencies that was produced in response to the Zika outbreak in 2016. So a number of bodies have sort of brought this to the fore again and reaffirmed their commitment. And of course, there's an ever-increasing number of databases that have been set up by a range of organisations, including the World Health Organization, UK Academy of Medical Sciences, within the US National Institute of Health Strategic Plan, and of course the European COVID-19 data portal. And all of this hopefully will not only progress research better, but will help to reduce animal use and avoid animal use. And of course, this is what this whole webinar is all about, avoiding and replacing animal tests. So some key points here are the global workshop that was convened by the International Coalition of Medicines Regulatory Authorities to critically analyse which preclinical tests can safely be avoided. and we'll be hearing from them on the second webinar tomorrow. There's also been some parallel testing in humans, which has helped to reduce the number of animals used. And of course, there have been some very exciting new developments in non-animal technologies that are predictive and clinically relevant. And I'm really looking forward to hearing more about those in this webinar. So where does all this leave us then? It's a desperate situation, but it's come up with both negatives and positives for laboratory animals. So what can we do? I think it would be really helpful if we could use this webinar to think about what we could all do within our different roles to achieve a positive shift in scientific culture and to keep the door open for the 3 Rs, to borrow Thomas Hartung's concept. So I'm going to talk about promoting the positives and being mindful of the negatives, and then just give some examples of what animal welfare bodies and IACUCs can do. So the positives then, within all of our working roles, whatever they are, if there are any ways to sort of promote these and champion them, I think that would be a very positive thing. So obviously it needs more funding for the development and validation of non-animal technologies and greater incentives to search for and use them. It would be great if this collaboration and data sharing could continue at its current levels to reduce animal use and duplication. And maybe regulators could convene more often to identify animal tests that could be avoided across the board, not just in relation to coronavirus vaccines. And also, wouldn't it be good if scientific impact could be measured primarily in terms of bringing therapies and treatments to the clinic rather than publications in high-impact journals? And the flip side is being mindful of the negatives within our particular roles. So, for example, it's important to challenge scrambles to use animals as the default approach rather than the last resort, which they should be, especially if severe suffering is involved. It's also important to call out anyone who is downplaying animal suffering or trivializing animal use, to call out research being speeded up so that it's no longer rigorous or high quality, and to challenge people who don't collaborate or share data. Also, there can be a risk that benefits are overplayed or the likelihood of achieving them is overplayed, which could skew the harm-benefit analysis. And we've also seen some factions extending the arguments made for using laboratory animals during this unprecedented global emergency to come up with generic justifications just for using animals in research and testing in itself, which is just maintaining the status quo, which is really not acceptable. Now, I'm going to just set out some thoughts for committees such as animal welfare bodies, IACUCs, animal ethics committees, and the UK AWERB, because this is something that I have a particular interest in. I'm sorry for the amount of text on this slide, but they're just all important points, and I just thought it would be helpful to have them all set out there. So if you sit on a committee that has a task of advising on the 3 Rs, it's really important to reflect on whether there are incentives to search for and implement all of these. And do staff actually know how to search effectively? Do they receive adequate training in this? All too often, people search for refinements and just tweak projects around the edges without really fundamentally thinking about all 3 Rs. Do the research teams collaborate to share data, approaches, and 3 Rs information? Do you and your other committee members actually have the necessary competencies to advise on searching for the 3 Rs? And then very importantly, when work resumes, will there be contingency plans in place to avoid killing animals if there's another wave of coronavirus? Because this led to some very difficult decisions and issues, animals' lives being wasted, and a huge amount of distress for animal technologists who had to bear the emotional load of killing these animals first time round. If the committee has a task of keeping staff informed of technical and scientific developments relating to the 3 Rs, are there actually adequate information channels, and is this task properly resourced? Certainly amongst a lot of AWERBs in the UK, the answer to both these questions is no. And then if there's a task of following the development and outcome of projects, are there processes in place to properly capture ways in which the 3 Rs have been furthered, and were harms to animals accurately predicted and alleviated effectively? So these are all things, obviously these are all important at any time, but I think we can use the coronavirus crisis to really charge these issues and encourage further reflection on them within the committees that we sit on. And this is really all relating also to the impact that these committees can have on developing the local culture of care and implementing this. And the culture of care is about more than just caring for animals. It should be an establishment-wide commitment to improving welfare and the quality of the science, care of the human staff, and transparency. And the last point on the slide here is really important. It's about going beyond legal requirements, not only with respect to housing, husbandry and care and experimental design and all the rest of these kinds of issues, but also in thinking about identifying and acting on wider ethical issues, because that's a really effective way to challenge an anything goes kind of culture if it crops up, and ensure that science is done humanely and responsibly and using the most up-to-date predictive technologies. And just a final slide from me, while we're considering wider ethical issues, this is kind of off scope for this webinar in a way, but it kind of escaped all of your attention that a lot of commentators have been making the point that animal and human health and welfare are intimately linked. So many disease outbreaks like this one, going right back to the so-called Spanish flu of 1918 and previously to that, have been associated with destroying the habitats of wild animals, eating wild animals, and with farming practices. And these are very complex issues. There are few easy solutions, but unless radical changes are made to the ways that humans use other animals and share the environment with them, these cycles of disease and suffering will continue for both humans and animals. So getting the scientific house in order and sorting out the life sciences is just one step towards dealing with this problem. And that's all from me. Thank you very much.
Thank you, Penny. I didn't even need to give you 3 minutes warning on that. You're right on 17 minutes. Super. We're starting to get some questions coming in. So if I can ask you to continue to contribute questions, to send them on the chat box. Our team will moderate those and aggregate them where necessary. Please send your name as well if you like, and we'll try and give you a shout out when we get to the question and answer section. Let's go to our next speaker. Koert Stittelaar. He is a PhD. He's a general manager at Viroclinics Xplore in the Netherlands. Koert, good to have you on board today. You want to switch your microphone on there? And you've got your 20 minutes, and the team will get your slides going for you. I'll give you a shout out with 3 minutes to go. The floor is all yours.
Thank you very much for the introduction, Brian, and I thank you for giving me the opportunity to speak. So I'm a virologist. Next slide, please. So I'm working for Viroclinics, which is a contract research organization. I'm based in Schaijk, where we have our preclinical facility. Next slide, please. So I'm mainly involved with the group performing early stage research and preclinical research. Next slide, please. So over the years, we developed quite a number of platforms to test countermeasures against respiratory viruses, so ex vivo and in vitro, and also animal models. So in the left table, you see that we have quite a number of models against influenza, but also other respiratory viruses, including coronavirus. And our main field of working is testing vaccine efficacy and antiviral efficacy. And to do so, you need the relevant readouts, like being able to look at viral loads. Next slide, please. So for us, working at COVID-19 started with that we were kindly provided with the virus by Christian Drosten from Berlin, Germany. Then we obtained the virus stock from a clinical isolate. We grew the stock on Vero-E6 cells. We developed a titration assay with an endpoint readout based on the use of WSD8, which is a fatality staining. We developed also the back titration so that we would be able to re-isolate the virus from tissue during infection experiments. And we set up with the virus neutralization assay. And in the first months of the outbreak, we worked on the models indicated on the slide. So we did experiments with ferrets, rabbits, and hamsters. We tested different infection routes, intranasal, intratracheal. We tested different doses. We also tested different sampling times and different moments of necropsy. And we set up, and where we could, we used that, the in vitro systems like human epithelial cells, so there are liquid interface cells were very valuable in our hands. Also organoids, intestinal organoids, and the vero cells themselves that were also already available for the first testing of antivirals. Next slide, please. So in this presentation, I like to use two examples of what we have seen in the past 10 years in terms of other emerging viruses. So the two examples that I like to use are the pandemic H1N1, which we all know as the Mexican flu or the swine flu, which emerged in 2009 in Mexico. It was a zoonosis from pig to man. And the point of reference that we had at that time were, of course, the circulating influenza viruses, but also the highly pathogenic avian influenza viruses that we already knew, like the H5N1. And for those viruses, so the circulating influenza and the high pathogenic, there were already models established, so like ferrets and mice. The other example is the MERS coronavirus, very relevant for this talk. It emerged in 2012, Saudi Arabia. It was a zoonosis from dromedaries into humans. And the point of reference there was the SARS virus that emerged in Hong Kong in 2003. And for that virus, also animal models were established. So first about the pandemic H1N1. Next slide, please. So it is a relevant example, I think, because among the different influenza viruses, we see that very similar viruses can use different receptors. And that's also that's what we now also know for coronaviruses, and that they can really be very important in setting up proper experiments. Like if you use the wrong inoculation route, like what's here now in the right middle, described, if influenza virus H5N1 is given intranasally into animals, it could rather develop a CNS disease than the severe pneumonia, which is the disease where we needed to develop countermeasures against. Next slide, please. So setting up proper experiments, so especially in the context of the three R's, because the quality should be good, it should be reproducible, the test should be robust. So there are a couple of parameters that are quite important. So that's the infection dose of the challenge virus, I mean. The infection route already mentioned, the readouts that we do, and when do we take the readouts. But it is also important that we know on forehand already, if it concerns, for instance, antiviral testing, that we know the pharmacokinetics, so that we know how to dose the candidate antiviral drug. And if it is about testing a vaccine, that we already know about the immunogenicity profile, especially if you talk about the use of adjuvants, because it might be that an adjuvant for human use is not performing similarly in a certain animal model. Next slide, please. So back to the dose, the challenge dose. So obviously, if you take a too low challenge dose, you could make it a poor vaccine, too easy to show efficacy in experiments that you run. And then at the end, you would not end up with a effective vaccine. On the other hand, if you would dose too high, then you could make it a promising vaccine too difficult in the testing and jeopardize this product so that it would also not reach the market. And that is also a not proper use of animals. So focusing on this red box at the bottom right corner, I'd like to indicate to you that there are parameters that are sensitive to a different challenge dose, but also that are not sensitive. So the example like macroscopic percentage lung lesions, that varies with that increases with increment of challenge dose. But the lung viral titer, which can be a very important readout for respiratory virus vaccines, that is on a kind of plateau, so that is not sensitive for challenge virus. Next slide, please. So the samplings that we take and for assessing these parameters that I mentioned, like the lung damage, the lung viral load, or are we doing that at the right moment? Next slide, please. So there are different ways to find out what is exactly the kinetics of the infection that we are studying. So here, still, this is the pandemic H1N1 as an example. So the upper row shows histology pictures from the lung. And if you use a number of animals, and that is the negative side of it, then you will be able to see over time where most of the histopathological changes are. And then you will be able to see where, for the particular question that you have, it will be relevant to perform a necropsy. The disadvantage is that you need a number of animals, and you are comparing data from different animals. And in terms of the 3 R's, I'd like to point out to you that there are techniques like the CT imaging, where if you use that, in principle, you can work with one animal and follow that single animal over time, starting at the baseline to show that at the start of the experiment, the lungs are healthy, and then you can follow the disease over time. Of course, we have to realize, like also the SARS-CoV-2, that's a B-cell3 pathogen, it's not always easy to implement such techniques. Next slide, please. So this slide I made 2 years ago, 2 years after the outbreak of the pandemic H1N1, just to indicate, I'm using that to indicate to you that if you look in the scientific literature, and if you compare what the different scientific groups did in evaluating countermeasures against the virus, so either antivirals or vaccines, the challenge protocols are very different. So different in duration, so they were either short or long. If there is necropsy involved or not, samples, certain types of samples are taken at different moments. So there is hardly any standardization, and I think also in terms of the 3 R's, it will be important to look at this standardization, because that would allow comparison and could also, in principle, avoid the double use of animals. Next slide, please. Also for proper and good quality of experiments and the discussions that we need to have with the animal ethics committees, it is a good practice to introduce go-no-go decisions. So here as an example for vaccine testing, a first experiment could involve the testing of the immunogenicity of a vaccine. Typically that could be tested like a prime boost, or maybe even with 3 doses, so prime boost with a typical interval of 3 or 4 weeks between the different doses. Blood samples could be taken for a serology analysis. But then next slide. If that analysis, so there is, for instance, the serology analysis or otherwise the responses that are relevant as a readout are measured online, so during the experiment, then in principle the same set of animals could be used also to address the efficacy. So to challenge them with the particular virus, to take the samples at the right times and perform necropsy if needed, if that is required to answer the question. But this is in principle a practice that could lead to a reduction of animals. Next slide, please. So for the pandemic H1N1, let's say we were good. So the established models could be used to also test countermeasures against this virus. But now for the corona, the SARS coronavirus that we see today, the story is a little bit different. So now I'm going further with this MERS coronavirus example. Next slide, please. So the point of reference was the SARS 2003 virus that emerged in Hong Kong. The postulates of Koch, so that those are guidelines to identify the causative agents. So really to prove that the isolated virus is indeed causing the disease. That was done in the Osterhaus group in the Netherlands using non-human primates. Next slide, please. A bit unfortunate. For SARS 2003 virus, it was discovered that it used the ACE2 receptor. It was found also by the group in Rotterdam, by the group of Bart Haagmans, that the MERS coronavirus was using a different receptor, DPP4, expressed also on a different set of cells. Next slide, please. So looking at the phylogenetic analysis of this DPP4 and the existence or the presence in the different species. So you see in the bottom that it's present in the bat, or the virus originated from bats, and on the top you see the humans. So in principle, that all the species on this slide could be in principle used as platform to test countermeasures. Next slide. An example on the previous slide, ferrets were included, and ferrets were also an established model for the 2003 coronavirus. But for the MERS, it was not a particular good model. So the ferrets did not seroconvert; they did not mount antibodies against the virus upon infection, and also the virus re-isolation was poor. And this was seen for different species, so the search for proper models had to continue. Next slide, please. In the search for proper models, so we used a non-animal platform, so using cells, expressing the receptor from the cells from different species, and then and also the receptor from different species. And these experiments with binding studies and biochemical analysis revealed that in principle the rabbits could also serve as a animal model for MERS coronavirus. Next slide, please. So that was true. So the virus replicated also in vivo, and the model could be used to study vaccines and antiviral drugs. Next slide, please. But once you have the model, there are you are obliged to see if you can improve the model to improve the quality of the research, and also as a refinement so that you can get more data out of the animals that you are using. So this is an example that I'd like to explain to you. Here you see a cartoon of the lungs from a rabbit. So our standard protocol was showing at the left was to take biopsies from regions A, B, C, and D, and to pool these pieces of tissue into one tube to make an homogenate and to show the presence of virus. But if we compared our findings, and sometimes the virus back titration was negative, if we compared that with the microscopy analysis, there was a discrepancy, and so we should have had a positive result. We changed our protocol, so that's on the right side. Instead of 4 biopsies, we collected now 9 biopsies and did not pool them, but keep them separate. And then next slide. And this allowed us to show difference between placebo, so unprotected control animals, versus the successfully vaccinated animals. So such a modification of the protocol could really also help us reducing the numbers of animals and to get more data out of the animals that we are using. Next slide, please.
Just 3 minutes to go. Okay, Koert?
Okay, I have to, so skip this one, please. Next slide. So for the SARS-CoV-2, that's depicted in the right lower corner. It's genetically more similar to the SARS virus from 2003 than that it would be from MERS. It is using the same receptor, so also the ACE2, So transgenic mice for ACE2 could be used to fulfill the postulates of Koert, to prove again that this is the causative agent. Next slide, please. Next slide, please. So here on the left side, you see the models that were established for MERS corona. In the top, below that, you see the models that were established for the SARS of 2003. So we needed to find the suitable models for the new coronavirus, for the COVID-19 virus. So first we did that with a non-animal solution, so human target cells transfected with the ACE2 receptor from different species to see whether these are sensitive for replication by the virus. Next slide, please. And the different groups around the world were looking for the proper models. So also the WHO had a weekly meeting to get together and to exchange the early information. A publication will soon be published showing the in vivo models that are listed here in this table. But there are always ways to, and there is a need, of course, to look for alternatives. So ex vivo can be a solution. So from one animal, you could take, for instance, the lung, and you could take lung slices and divide it over different test cups. So then with one animal, you can perform maybe a whole experiment, including the controls. And then in vitro, there are, again, I already mentioned them, the use of vero cells, other cell types, organoids from humans and from animals. And below the table in the right side, so as mentioned, scientific literature is important, also to share results and to avoid the double use of animals. The big data analysis is a way to prevent using animals. But also at this moment, I just like to raise the question, is this maybe not too early to have access to alternatives? Because first you need to know how you can test it before you can find an alternative for the particular test. I will get back to that. Next slide, please. So there are now a number of animal species that could be used for COVID research, but which animal to choose? So there are in principle, the question determines the choice. So what do you like to address? Is it maybe the longevity of a vaccine? In that sense, then the lifespan of the animal species that you are using may be relevant. And there is the question, so there are multiple models, why not just one? Well, I think that is because of each model has its own feature. Next slide, please. So in this discussion, so the question determines the choice. So what reagents are available? Is the pharmacokinetics proper in the species that we are using? Is that similar to what we see in men or what we expect in men? Existing data may drive a certain choice. And also the application. An interesting example is that we heard from the Netherlands the infection of mink. So in principle, minks could also be used as a laboratory animal, and then you would use it as a model to develop human countermeasures, but also maybe for the species itself. So to serve. Okay. So this is in the One Health phenomenon. Next slide.
Just 1 minute to go to wrap up then, okay?
Okay. Next slide, please. So the three last slides to indicate, I think, what is important and that is recognized by others and where it's probably unavoidable to use animal models. So one is vaccine safety, so vaccine-induced enhancement of disease. Next slide, please. I think also important, if you look at the numbers and reasons why people can have a deficient immune system, if you count all these possibilities up, you may reason that about 10% of the human population is somehow immunocompromised. And it's in principle what we see. The weaker people in the population that are showing more mortality and morbidity from COVID. So this is important. This is difficult to test in people. So here we may need an animal model. Next slide, please. The animal model is there, but I skip it for the sake of time. Please, next slide, please. Transmission studies are important. Probably also very difficult to replace that with a non-animal solution. Next slide, please. And so I'm coming to the conclusion. So I've talked about the development of the models where we can use alternatives, and about the existence of different models and reasons why to choose for one species or another species. I think further validation is needed, especially also for replacement. So is a replacement really an alternative that needs to be proven? At the bottom of this slide, a subject that I didn't mention, and I think very important to include it in future experiments, is the pre-immunity. So if there are now people that are already immune to the virus, are vaccines effective in this population? And how about protection against reinfection? Next slide, please. Yes, I reached the end. So these are the acknowledgements. Thank you for being patient with me.
Thank you, Koert. That was excellent. Okay, thank you so much. Our next panelist today, Christian Desaintes. He's a policy officer at the European Commission, just down the road from me here in Brussels. Christian, you have 20 minutes. I give you 3 minutes head up when time comes. The floor is all yours.
Okay, thank you, Brian. It's a pleasure for me to be at this webinar and to give you an overview of the research response that the European Commission has provided to address the COVID-19 pandemic. So the European Commission has reacted very quickly by providing ambitious actions in different areas, including research, for which more than €1 billion has been allocated to COVID-19. It all started on 30 January with the first expression of interest, from which 18 projects were selected. 7 projects on epidemiology and public health, 3 projects on diagnostics, 6 projects on treatments, half of them on antibodies and the rest on antivirals and sonatides. 2 projects on vaccines, but really the early stage development vaccine. One vaccine is a DNA vaccine against N protein, and the other one is the VLP. So in total, it's almost €50 million. 3 March, there is the Innovative Medicine Initiative Fast Track Call, which selects 8 projects, 5 on diagnostics, 3 on treatments, including antivirals, but also clinical trials on TK inhibitors for acute respiratory distress syndromes. So in total, there were 72 million from the European Commission and an additional 45 million in kind from the pharmaceutical industry. 19 May, there is a second expression of interest. The evaluation of proposals has recently been concluded, and the 23 projects are shortlisted for a total amount of 128 million euro. Among these projects, 2 are on cohorts, 4 on repurposing of manufacturing for vital medical supplies and equipment, 13 projects on medical technologies, digital tools, and artificial intelligence to improve surveillance and care, and 4 projects on the impact, either behavioral, social, and economic impact of the outbreak response. So next slide. So 13 March, there was another call from the European Innovation Council, which resulted in 36 projects being funded for a total amount of 166 million euro on testing, treatment, prevention, including vaccines, but also disinfectant and data analytics, and others, including remote care to patient, quality control of coronavirus-related project. On 3 April, there is the EDCP call, for RNI and infrastructure in sub-Saharan Africa. 20 projects were allocated to natural history of infection, point of care diagnostic test, trials of promising products, whether diagnostics or therapeutics, and then finally validation of serological tests and the establishment of biobanks, including for sera of COVID-19 patients. So apart from these direct actions, the Commission has also provided generous support for vaccines. 100 million have been given to the Coalition for Epidemic Preparedness and for extending the portfolio of the CP projects and the global manufacturing capacity. Also, 75 million have been given in forms of a loan to Euractiv for proceeding its mRNA vaccine. 20 million euro have been provided to collect convalescent plasma, but also extending the discovery clinical trial, which was initiated in France. 15 million euro were devoted to infrastructures. 7 million euro were given to the health cluster of the European Institute of Technology, which allow funding of 15 projects on diagnostics, digital health, and medical technologies. And finally, 325 million euro are available from a collaboration between Horizon 2020 and the European investment banks in forms of loan in the instrument that is called INOFIN, Infectious Disease Financial Facility, to allow companies to fund products which are relevant to COVID-19. So what about the animals in all these projects? Now, if we take the running COVID, well, some of the project already started a few months ago, but if we take all these projects either running or which will start soon, there are more than 100 COVID projects. It's more than 500 million euro. And there are only 10 of these projects which use animal studies, which is not very surprising because many of these projects didn't really need animal studies. But even in these 10 projects with animal studies are usually only a limited part of these projects. So I think that it's safe to say that less than 0.2% of the budget or the resources are just provided to animal studies. There are 3 of these projects which use non-human primates, and these are all projects dealing with the treatment. One project is also using wild animals, but this is only for looking at species of bats and wild mice to follow the evolution of the virus. Now, what about the alternatives to animals? Well, not very surprisingly, there is also 10% of the project which use alternatives to animal studies, because most of the projects not using animals wouldn't need to use alternatives either. But the good news is that the project using alternatives to animals have already generated results. So in a way, they generate quick results. Among these projects, among the alternatives, there is the air liquid cultures of human airway epithelium. One project has generated 17 human antibodies with the phage display technologies, and these antibodies are neutralizing antibodies interfering with the SARS-CoV-2 receptor binding domain of the spike with the AC2, and they prevent virus infection. And you can see on the slide on the right above, you see all these 17 antibodies, and then you have below you have 4 columns of cells The ones at the right are the cells which are not infected. Next to them, on the left, these are cells which are infected, and you can see that the density of cells is lowered. And then next left, you see that the density of the cells is returning to normal with one of the best of these 17 antibodies. Now, another project is using supercomputers, artificial intelligence, and biological processing to screen millions of molecules. And the idea is really to model the interaction between all these molecules and the viral proteins. And it has already screened 400,000 molecules. And this led to the identification of one molecule, which is a haloxyfen used in the treatment of osteoporosis. And this molecule is a potential effective treatment for COVID-19. Now you see that, the Commission has provided a very substantial research contribution for COVID-19, but also the Commission is a strong support of the development of alternatives to animal testing. Over the 20 last years, the funding has been more than 700 million euros, and probably that at the end of Horizon 2020, this number would probably increase to more than 800 million euros. So we are in a way close to the 1 billion euro that is provided for COVID. There are more than 200 projects in total. Now, as you can see also in the graph on the left, the annual budget for alternatives has increased through the different framework programs. It was 11 million euro per year in FP5, and this amount tripled between FP5 and FP6, with an additional 50% increase from FP6 to FP7, and then it remains relatively stable from FP7 to Horizon 2020. Now the question is, what did these projects on alternative contribute to COVID-19? Well, I just contacted all the coordinators of these projects, and there are more than 75 projects, more than 250 million euro. And I've asked them a few questions. A third of them already replied. It was during the holiday time, so we might get more reply in September. But basically, the idea is that 35% of this project redirected some activities toward COVID-19 research, and more than half of the project contributed to the tools that I'm sorry, there is more than half of the projects contributed through the tools that they have developed to COVID-19 research. And I give you just some examples. One project developed microfluidic devices with the stem cell-derived human vascular endothelial cells to model COVID-19 associated thrombus formation, and adding immune cells to model the inflammation. Another project built a brain organ on a chip. Another model developed an air liquid interface cultures of nasal epithelial cells, and which recapitulate actually the expression pattern of AC2, such as is observed in vivo in goblets, secretory cells, and ciliated cells. So in principle, this is a very good model with a totally differentiated model. A number of projects developed organoids, such as the organoids for small intestine. You can see on the picture on the right, this is the small intestinal organoid, which is infected by COVID, by the SARS-CoV-2 in green, and the virus can replicate in the enterocyte, and there is a viral response which can be monitored in these organoids. Liver spheroids were also developed to study the effect of a JAK kinase inhibitor, which might be used in treatment. Lung organoids, obviously, and then cardiac organoids to examine cardiac damage, and also kidney organoids that you can see here at the bottom, where you see in green the tubule-like cells, And these organoids can be infected by SARS-CoV-2, and the infection can be inhibited by soluble forms of human recombinant ACE2 receptors. Now, other alternatives include human primary lung cells, which was used by one project to show that SARS-CoV-2 prevents burning carbohydrates, which result in the accumulation of fat in the lung, which is required for the virus to replicate. And the project is currently testing a cholesterol-lowering drug, fenofibrate, to see whether it can be of any use for COVID-19. Other projects develop differentiated IPS into upper airways. One project in collaboration with Xplore GRC is trying to link the AOP, which was developed for lung toxicity, and trying to build an AOP or link it to lung disease AOP or a COVID-19 AOP, if you want. Another project developed a procedure for biocoating the spike protein, and this is a way to screen drugs and siRNA. Another project uses zebrafish larvae for cardiotoxicity of hydroxychloroquine and other drugs. And finally, there are models for distribution of AC2 in different human tissues and organs. But basically, you see that these projects cover basically all the different organs which are known to be targeted by the virus. Another point which was already alluded to by Penny was the European COVID-19 data platform, which contains 3 components, of which one is the COVID-19 data portal, which allows the quick sharing of information, including sequences not only from virus but also host, gene expression, proteins, biochemistry, pathways, and finally a huge amount of literature, as there are more than 140,000 papers. Now, what is the effect of the pandemic on the, I'm sorry, next slide. Yes, next slide. Okay, so now what is the effect of the pandemic on the use of animals in this year? So we will, in Europe, we will be able to have details on the number of animals used for research, but not only before 2022, because the member states have to report to the Commission by the end of 21, and then the Commission will issue its report hopefully by the end of 22. So it's not only before this time that we will know about the number of animals, but then what about the animals which were bred and killed without being used for scientific purposes this year? Once again, but again, we will not have this information since this information is required only every 5 years. The last time this information was required, and actually also the first time, was in 2017, and so we will have this information only for the year 2022. So next slide. But what is the effect of COVID-19 on animals beyond 2020? And of course, maybe this is the right time to reflect on the future role of test animal in science. And okay, when I contacted all the coordinators from the alternative projects, I asked them exactly this kind of question, but I asked the same question to all the coordinators of the COVID-19 projects. And the opinions differ slightly between the two, let's say, the communities. For the alternative communities, 20% of the coordinators say, yes, this is the right time maybe to reflect on the new role of test animals. But, majority of them were just pushing for an increase in animals testing. What for the COVID-19 community, basically people were not aware or didn't have very strong opinions, because mainly these are people not using animals, and they probably don't care about the use of animals in science. But what was clear is that both communities are strongly against a ban on animal testing. But the European Commission organizes a conference in Brussels this year on 10-11 December, and this, I think, it's the right maybe opportunity to get a reflection on the use of test animals beyond this crisis. Next slide, please.
3 minutes, Christian.
Okay,
minutes to go.
This is the last slide.
Okay.
So as a preliminary conclusions, I would say that, the Commission is really a strong supporter of research for COVID-19 and alternatives, because both together we are close to the 2 billion euro, Then there are several projects have generated alternative tools that were immediately used to the COVID-19 research. And within the new COVID-19 research projects, we have seen that some of them using alternatives have already generated quickly interesting results. But also, some of the Horizon 2020 projects have built up a comprehensive set of methods for really quickly testing the safety of new drugs. But the tools they've developed were not taken up by the pharma industry. So I think that we should try to analyze why, and we should try to see how to better, communicate between the different communities, and maybe how to better advertise, the availability of such nice tools. Also, you should realize that when we mention alternatives, it's not always understood the same way by all communities. Even in this community, when we talk about alternatives, sometimes it's alternative, sometimes it's NAM, sometimes it's NAD. But for the COVID-19 people, when you talk about alternatives, I'm not sure they understand really what is alternative. So I think that there is really an improvement to make to try to speak the same languages. And then finally, it's not always easy to identify the COVID-19 research activities using alternatives. And I will take the example of the monoclonal antibodies, which are going to be used for therapeutic purposes. Around the world, there are more than 40 different therapeutic programs for monoclonal antibodies under development. And the majority of these antibodies were developed from mice, and very few on phage display. And then there is the difficulty to find this information, how they were derived. And then it's very interesting to see that despite the fact that GRC has produced a very interesting report early this year on the validity of this phage display technique for generating monoclonal antibodies, still when you see the programs in the world, very few are using this new technology. So I think that we need to understand better how to maybe better publicize the fact that alternatives are there and they should be taken up more. So I think that we need more crosstalk and better information on existing alternative methods. And with this, next slide, please. I would like to conclude and thank you for your, thank you for your attention. And next slide. I'll be ready to take any questions. Thank you. Over.
Thank you. Thank you, Christian. Thank you to all our panelists. We have lots of questions coming in now. I encourage our audience to continue sending in questions. We will take them as many as possible. We have over 400 people online from all across the world today. And so thank you for taking the time. I appreciate you're not all on the same time zone that we have. So if you're just joining us for your morning coffee, you're especially welcome. Let's get straight to the questions. I'm going to take them pretty much in the order in which they came in. One for Penny. This is from Pauline Yurkov. And Penny, the question is, many people use the pandemic as an argument for the need of animal research and against initiatives to replace or reduce animal use. How do you feel about this trend?
Oh, hi, Pauline. Yes, I take a very dim view of this trend because it's politicizing a very desperate situation when actually, if you think about it, it's kind of an extension of business as usual. So this situation is very desperate, but you still have a situation where there's an unmet medical need. There's an array of different approaches that are being used to meet this need. As I said earlier, it's impossible to predict the relative contribution that each of these different techniques is going to make. So if you're going to isolate animal use and champion that as being essential, well, you can say the same thing of epidemiology, computer science, organ on a chip, all of the other approaches that we've been hearing about. So it's not helpful, and I think it's a bit distasteful to seize on this push an agenda when actually it should be about doing the best quality, most humane science. And as we've seen, non-animal technologies are certainly coming to the fore. So I just think it calls for a bit more of a sense of proportion and propriety, really.
Thank you. Good summary. Give this next one to Koert. Not specified for anyone in particular, but you spoke more about animal species. So, Koert, what makes you choose the 3 animal species used in testing corona, for example, rabbit and the root versus species? Maybe you want to expand a little bit on what you said earlier.
Yes, rabbits are indeed amongst the panel of species that could be used. So it's still at a relative early stage. So we are talking now about, let's say, 7 months of research, and the research is taking its time. It's not a one-day experiment. Around this time, really all the data is coming in, and we can compare and see what the features of the different models are. We see now that rabbits, they are shedding the virus. The rabbits are not becoming sick. They are not showing clinical disease. Studying rabbits is now important because there is also a concern: what is the role of rabbits in the transmission of the disease? There are quite a lot of rabbit breeds in the community. It's not a disease model as in its standard way. One of the concerns that I refer to is the vaccine enhancement of disease. That is really what we have seen in different viruses, like with dengue and RSV, where a vaccination may not be safe. So if you like to see enhancement of disease, there must be room for enhanced disease. So if you are already working with a disease model, then you may overlook this phenomenon. Their rabbits may have a value to address this topic.
Okay. I think a phrase used earlier was the question determines the choice. I think it was a neat summary of that as well. Let's go to our next question. Thank you, Koert. This one goes to Penny, but I'm going to give it to all of you. Maybe you're all aware of different issues with this. No name for this one. Are there any case studies of research not conforming to current legislation and regulations and overlooking the use of animal-free methods in COVID-19 research, or case studies of research not properly implementing the 3 Rs due to the COVIDization of research? Christian? Penny?
Maybe I will start by saying that all the projects that are funded by the European Commission undergo a very thorough ethical review process where the researchers have to comply or have to demonstrate that they comply with the 3 Rs. So I'm not aware of any research project not conforming to the 3 R. Over.
Penny, Koert?
Would you like me to go next?
Yeah, go ahead.
I would answer this really with reference to the ongoing crisis within the life sciences with respect to reproducibility and translatability. So notwithstanding all the review processes that are in place, there are still a lot of scientific studies that are published that are not properly randomized or blinded and that don't include enough information when they're reported. And I think this is likely going to be exacerbated by the kind of desperation science that I referred to earlier. So while there have been no publicized cases where people have used animals when alternatives were available or where the 3 Rs weren't adhered to, I think this is likely to be happening because of the reason that I explained earlier, because of the ongoing reproducibility crisis, and also because of the lack of available information about alternatives lack of incentives to use them, lack of training in recognizing and implementing them, and also this kind of toxic scientific culture in which there is a huge amount of pressure on researchers to keep generating papers using the same techniques that they've always used, because there's this disincentive to learn new techniques and to change approaches. So I think that's an ongoing problem, but this pandemic is likely to make things worse in all counts.
Okay, thank you. Koert, let's continue on with the same theme. Just another question on the tail on from that, from Bea Zur. Bea asks, are there examples of cases in which the ethical evaluation has been less critical to support fast start of COVID-19 animal research? And does that also apply to clinical studies? What do you think?
Not that I'm aware of. So I think the system in the EU is very good, is very stringent. So the animal ethics review is good in the sense that we are obliged to use alternatives if they are available. If a certain question to us comes to perform an animal experiment, we simply start always with the discussion, is it really needed? And are there no alternatives? And if there are no alternatives in our laboratory, then we would direct them to a laboratory which is working on a alternative. So not that I'm aware of. On the clinical, I cannot comment, I'm sorry.
Okay, thank you. If anybody else wants to jump in that later, feel free. But let's go to another question. Christian, give you this one first. What's your scientific objective or objectives used to define a successful model of COVID-19 infection? You want to have a go at that, or you want to pass it?
Is it for me the question? Or?
Yeah, go ahead. Can you take that question? Christian.
Yeah, well, I guess that it depends on the publication and the outcome. At the Commission, we are not judging ourselves. The scientific.
But you're funding the project, so you must have some idea of what a successful model or successful outcome and according to the criteria would look like. Are there any particular characteristics you would highlight?
Well, we are relatively bottom-up. We use the approach of bottom-up, so we don't always define very precisely what we want, but we leave the freedom to the scientists to come up with the best models. And all the proposals are evaluated by external experts, and the best projects are selected based on the opinions of the experts. But of course, we have some ideas of what the best models would be. It would be.
Well, that's what I'm asking you. What would that look like? Penny, go ahead.
Right. Yes. Just thinking about one of the points I made earlier, and I said, wouldn't it be nice if research output was judged according to treatments being brought to clinic rather than publications in high impact factor journals? I think that would surely have to be the ultimate test of a successful animal model, or any kind of model, whether it actually brought something to the clinic. And that's a really good case for proper retrospective review of benefits of animal research to see whether it has actually delivered what it's proposing to deliver on a large scale.
Good point. Koert, anything to add to this?
To answer the question, also to comment maybe a little to Penny. So somehow the test platforms and animal models, they also serve as a filter. So it's not that everything that you test gets positively through the test. So the test is not done for nothing. The test result may be negative, and that is of high value. So there, a poor vaccine or an antiviral that is not doing the job should be filtered out of the system and should not be continued further.
Okay, excellent point. We have a few more questions here. Next one is, anybody can take this. Why do we still talk about alternatives to the gold standard, in inverted commas, of using non-human models when we should be talking about human biology-based and thus human-relevant research? And that's from Catherine Herman in the United States. Penny smiling most. Penny?
Yeah, just total agreement. Yes, why should the gold standard be something that is a model of something else? That makes complete logic, and I completely agree with Catherine. Good point.
Okay, any other quick responses to that? Christian, Koert? Otherwise, take another question. No? Okay, next question. Which cell models for SARS COVID-2 infection also model for inflammatory responses? Not sure who wants that. Koert?
Yeah.
Okay. It's
a difficult question, but I think that also the model mentioned by Christian, so the human epithelial cells. So in principle, the epithelial cells, if they are infected, they show a response, and that can be a cytokine response, and those cytokines can be analyzed. So that is at least a part of the inflammatory response. But yeah, it is not an intact organism. There is effort put into enriching organoids and primary cultures. So it is in principle possible to add inflammatory cells into this system, to add NK cells into intestinal organoids. So then you have you need to combine different culture systems, but there are possibilities.
Okay, Christian, anything to add to that? No? Okay. No compulsion. Let's another question. As we move towards a world with greater... Tell you what, if you're sending other questions and you don't want to put your name, just tell us where you're from. So it's a little interesting for me to find out. As we move towards a world with greater use of alternatives, what advice do you have to younger viewers who are starting university or first jobs, and how can they make concrete efforts to contribute? Your question. Penny.
Oh, that is a good question. I think the best way would be to seek out appropriate mentors. So, for example, to approach centres of excellence like Johns Hopkins and the Beast Institute, and to seek mentors within there and look to collaborate with them and to gain information from them. And I would also say within any sort of training programs that you receive, to ensure that you're being properly instructed in how to search for alternatives and how to think about alternatives with an open mind. There's far too much ticking of boxes going on when applicants are asked to demonstrate that there are no alternatives for their animal use. I think if people are given a proper grounding in how to search for alternatives, because it's not all, there's no sort of one-stop shop to look for alternatives. There's quite an art to it. So I think if they ensure that they receive the proper guidance that they deserve early in their careers, that should set a firmer foundation for them being able to contribute throughout their careers.
Okay, registering to vote as well is a good contribution to society currently for the science community. Christian, anything to add to that? Advice for young viewers who are starting out?
Yeah, I think that in Europe we have the 3 R's, which can be part of university curricula, and I think that we have 3 R's centers in Europe, and I think that we should probably expand these to maybe more universities. And we have also projects on the teaching, having a teaching program on the 3 R's, especially for students in medicine. But we should certainly maybe try to increase this.
Okay, Christian, stay there. I've got another question from Canada for you. Roberta Staley from Canada asks, please explain how the drug for osteoporosis came to be identified as a possible COVID-19 drug.
Can you repeat the question?
Sure.
I think that...
And just to add, if anybody else wants to send a question, please do it right now, because we're going to wrap up in 5 minutes. So, please explain how the drug for osteoporosis came to be identified as a possible COVID-19 drug. Do you know the answer to that?
No, it's not my project.
Try Google. Koert, anything to add to that? No? Okay. Just waiting for the team to send me through another couple of questions. In the meantime, let me ask each of you, what are you expecting in the next year's WC11 Congress in Maastricht? Will COVID-19 be the big topic then that it is now? What do you foresee for next year? Penny?
Well, sadly, I fear that it will be. We've been hearing a lot of reports lately to the effect that this is not going to leave us. It's not going to go away, and there are certainly going to be more regional outbreaks. So it doesn't appear to be the case that somebody will just develop a vaccine and it will all be over. I think there will be a lot still to discover and to achieve in order to reduce the virus to a level that the human race can live with. And sadly, I think other pandemics will certainly follow unless the human relationship with the natural world is completely rebooted, really. So I think pandemics in general, and this one in particular, will continue to feature for the foreseeable future.
The rabbits will outlive us, we expect. Koert, what do you think next year? It's going to be totally COVID dominated, or do you see anything else on the horizon?
No, I completely agree with Penny. So what we may see, but that's completely unclear. But at the moment, it is like I said during the presentation, it is a BSL-3 virus. So that limits us somehow in possibilities. So that means that everything has to be done in high containment. So besides the training that we require to work with animals and with different test systems, also it requires proper training to do this under this high containment. It might be that in the coming year we can see, like with the Mexican flu, that in a year time it is downscaled to BSL-2. So and that may enable us to apply more alternative techniques into the field.
Thank you, Christian. Final word.
Well, I think that we have seen an unprecedented increase in knowledge and sharing of knowledge, which was really never seen before. And I think that in the next year we will certainly take advantage of this new knowledge on the virus, which is very comprehensive, and new knowledge about a lot of different things, and also different actions, like, for example, the sharing of data. I think this is a good example, and this could certainly be applied to other domains. And I think that COVID-19 would probably be a milestone in the history of science. And I think that we will probably think differently in the future. And I think that 2020 will be the year where there might be change in our minds.
Thank you. For me, I'd love to hear more about supercomputing and AI in this mix as well. Thank you for your contributions today. We're going to wrap up here. Just to remind you that we're back on again tomorrow, same time, 3 o'clock Central European Summer Time. It's currently raining where I am, no evidence of summer. And you can use the same links. There's a different link today for Get Through to Blue Jeans, and for the link you can get all the information on the website. Thanks again to the Netherlands National Committee for the Protection of Animals, used for scientific purposes, for their support today. See you all tomorrow at 3 o'clock here in Brussels, and thank you for your attention today. It's much appreciated. Thank you to our panelists. Good afternoon.
Several years ago, the NCAD published a vision for reducing the use of animals in procedures, with the ultimate goal, a dot on the horizon, of achieving a worldwide shift to research that doesn't use laboratory animals. That dot has triggered quite a response. As a scientific committee, we are aware of the vital nature of intrinsic motivation, time and funding. Yet, we also know that the true innovation often comes from unexpected quarters. We are convinced it helps to promote those unexpected innovation by sharing target situations for research methods that don't employ laboratory animals in specific research domains. This includes objectives and action plans. The use of animal-free innovation is the central focus, which in the Netherlands is arranged through TPI.
TPI connects a broad range of stakeholders such as scientists, regulators, funders and NGOs to accelerate the rate of innovation. On TPI TV, scientists exchange ideas and knowledge, and the community can help you to answer challenging questions. Do take a look at TPI TV. Maybe you will even find things you didn't search for but which turn out to be very useful.
Our goal is to make the trophy of animal-free research a challenge cup that will be passed around by scientists in every field of research, in every country. Sounds exciting? Contact TPI.